FASTA format is just a plain text file format that we can use to store the sequences of proteins and nucleic acids. All you have to do is take a sequence in the conventional one-letter abbreviations for amino and nucleic acids. And what you do is you add a caretted header line that has information about the sequence. So maybe it has information about the species and the gene name. We'll get back to these headers in a minute. They can be super complicated, like if you take them from a database and they have a bunch of metadata in them, or you can simplify it to make it something that's simpler for you. So you can take any sequence and make it into a FASTA sequence, or you can get the sequence from a database such as UniProt or GenBank, and this is going to then have more information in that header line. Sometimes that information can kind of get in the way when you're trying to do things like align them and tell what's what. So I'll show you how you can just go ahead and change that header line to make it something that's going to make it easier to work with. So today I just want to show you how you can get these FASTA data files, as well as the basics about what the header lines mean, especially for working with UniProt, so when you're working with proteins. And I'll point you to resources you can use when you're trying to figure out what the different header lines mean for your nucleic acid format. So Wikipedia actually has a really nice guide to this, and you can see what various things in the header line mean. So let's step back and think really quickly about what FASTA actually stands for. So FASTA stands for fast all referring to that it is a software program that can be encompass both FASTN, which is nucleotide alignment, and FASTP, which is protein alignment. And so FASTA is this software, but FASTA format is this conventional file format that can be used by all these different software programs. And so I'll point you to the original literature. If you're wondering what the FAST stands for, I was too, and from what I can tell from online searching, it looks like it's not an abbreviation or an acronym or anything like that. It's just stands for FAST because it was a fast way to align these sequences. And so FASTA is this format that is then based off of how it was formatted originally was for this FASTA program. At least that's my basic understanding. And so let's go a little deeper into this format. First of all, where do you actually get this format? And so that's going to depend on the kind of data you're working with. If you're working with protein data, you're going to be using UniProt. If you're working with nucleic acid data, then you're going to be using something like GenBank, which is part of the National Library of Medicine's NIH's NCBI. And so more on those elsewhere, but I'll show you the basic ways you can get these FASTA information, as well as what it means. You can also, if you have any sequence, as long as it's in that proper sequence format, you can go ahead and just stick a caretted header bar on top of it, and then that will make it into a FASTA format. So I'll show you where you can get the FASTA data from the databases, but first let's go into the basic format. So again, you start with that caretted line that has the information about the sequence, say. It's also going to have information about databases that the sequence came from. Often that database information is going to be separated by this vertical separator line. And so here you can see different data, different codes will be coming from GenBank, will be coming from EMBL, SwissProt, et cetera, and those will be kind of separated by this vertical bar from sort of accession numbers, and we'll get back to this in a minute. But again, you don't have to have all that special information, that fancy information, you just need to have a caretted header bar. The caretted header bar is important too because it's going to differentiate sequences from one another. So you can actually have multiple sequences back to back as long as they're each separate, they'll each start with a caret, it'll be able to know that these are separate lines. So you can see here I have the vertical lines that are separating the database from the accession numbers and things like this, as well as then I'll show you this is information about the sequence, about the protein evidence, about the version number, the gene name. You'll see that this header line needs to be a single line. And so right now it's showing it's like multiple lines, but that's just because it's wrapping the window. You don't want to have any hard breaks in the return lines, new line symbols in that header line. That needs to be a single line. You can have breaks in the sequence. And so typically what you're going to have is your sequence is going to be the rows of your sequence are going to be 60 to 80 characters long so that it doesn't get really overwhelming on your screen and things like this. It's kind of convention that you keep them to those 60 to 80 characters. And it'll depend too in part on the program that you're downloading things from. But you can have new line characters in the sequence, but not in the header line. The return lines can make it easier to read the sequence on your screen, but it can also make it harder to do various things like search for strings within the sequence. So if you want one long string, what you can do is just copy and paste the text into a converter tool like this free website and remove the line breaks. There are ways you can do it with coding, but this is just super simple. What you want to do is convert the line breaks to none, convert the paragraphs to none. So if I go ahead and convert this, now I can just copy and paste, and it's a single line. It doesn't look like it's a single line right now, but if I scroll it, you can tell that it is a single line. I don't know why it's doing that weird thing in my editor, but it's a single line. That sequence has to be in IUPAC, IUB format. So IUB was the International Union of Biochemistry, which then became the International Union of Biochemistry and Molecular Biology, the IUBMB, which I've been an ambassador for and is a really great organization. So check out the IUBMB, but they have this nomenclature committee and things like this that basically standardize the abbreviations for nucleic acids and for amino acids. We have Dr. Margaret Oakley Dayhoff to thank for those one-letter abbreviations for amino acids. So she's considered by some to be the mother of bioinformatics, and she developed this one-letter code for the amino acids in order to make computational things with the protein sequences easier. And so I'll point you to some resources about her, really amazing scientist, and really important to know this one-letter amino acid abbreviations. What's really cool too is I learned that according to this article, at least, Dr. Pearson, and so Dr. Pearson was one of the scientists who developed FASTA. He collaborated with researchers in the Dayhoff lab when developing the FASP, which is the protein version of it. And so that's pretty cool, and I'll point you to these articles. The amino acids are each going to be represented by their conventional one-letter abbreviation. X can stand for any amino acid. When you're talking about nucleic acids, you're going to have an N for any nucleic acid, and then proteins, you're going to have an X for any amino acid. For both of them, you'll have a dash for a gap. Now that gap can be saying it's like missing one residue, or it could be missing 10. And so it's indeterminate length, so it doesn't tell you anything about the length of the gap, it's just that there is a gap there. For proteins, you're also going to have the asterisk for a translation stop if you're dealing with the translated nucleotide sequence. For nucleic acids, there's also some of these kind of more wild cards. And so in addition to the ACTGU, which you're all probably familiar with, there's also things like R, which is for A or G, so your purines, or Y, C2U for your pyrimidines. There's also K for G, T, or U, the bases which have ketone groups. M for A or C, the bases with amino groups. And then there's some other ones that I hadn't even heard of, but these more sort of wishy-washy ones that can stand for multiple things. But typically you're just going to be dealing with this like ACTGU. And then the standard amino acid letters. You'll often potentially see these N's, see these X's, and see those dashes. But that's the basics of that. In terms of the file formats, there's no standardized ending. And so a lot of these FASTA files are just going to end with .fasta, .fas, .fa. Those are sort of the generic FASTA endings. You'll also see things potentially like FNA, FFN, FAA, various endings that will specify what type of data is contained. But all of these are just plain text files that you could open with any plain text editor. And so I'm showing it here in Sublime, but you can also just open these in standard text edit. You can just copy and paste them into some sort of document. Sometimes that's all you need to do. And so this FASTA format really sounds a lot scarier than it is. It just got that carroted header line and then that sequence in the proper format. And by proper format, I just mean that it has the standard abbreviations for your amino acids or your nucleic acids. And you can make anything that header line. But the header line, when you get it from a database, is going to have information in it. So let's take a look at that information. I'm going to focus mainly on the information for the proteins, but for us, I will also show you just kind of where you can get the information for the nucleic acids. And so again, the Wikipedia article is really great about this. You can also go to the NCBI. They have information about this. Depending on where the sequence is coming from, the FASTA download is going to be in different locations. So here you can see I'm on this GenBank entry. I can go here to, if I scroll down here, you can see that it kind of has some sequence information. It's all broken up. If I go to this, I can change this to FASTA. Now I can see that FASTA sequence. If I wanted to actually download it, so I could just copy and paste this into something. If I wanted to actually download this, all I had to do would be to collect file. If I wanted to download the file, I could clipboard it and just copy it to my clipboard, or I could send it to a file. And so here, this is it from the GenPep database. And so here you can see that I have the sequence data down here, but it's not in that FASTA format. However, I can do this. I can go and I can change this to FASTA and get the FASTA sequence like this. And then I could also send it to a file just like before. So that's nucleic acid. One other thing I want to say about nucleic acid is there's also a FASTQ sequence format. And the FASTQ format actually has sequencing data information. And so this is just a simple one from when I had a plasmid sequenced. And so you can see that it starts with an @ sign instead of a carat. And it has these two sections separated by a plus sign. You have so the @ sign line is going to have information. Typically, this information is going to be about the sequencer. And so I'll show you I'll point you to guides, but basically you're gonna typically have information in that line. If you're using like Illumina, it'll have information about the machine and the lanes where it was sequenced. This one's a more simple header line, but you can see it still has those two sections separated by a plus sign. After the plus sign, this is going to be information about the quality of the read. And it has these letters that are mapped onto the probabilities or the quality Fred scores. And so it's an integer value representing the estimated probability of error, and then they map those to actual alphabetical letters and things. And so much more on that here. I'm not going to go into that, but when you have a FASTQ sequence file, that's what that's going to be. And so if you're dealing with sequencing data, you're going to have those FASTQ files. Often those FASTQ files are going to be really, really big, and so they're actually going to be compressed files, and so it'll be like FASTQ.gz or something. And so I dealt with a lot of those files when I was doing ribosome profiling in my postdoc. Not going to deal with those now. I'm going to deal more with the proteins. So when we're dealing with proteins, you're often going to be working with UniProt. I'll take a second to talk about what UniProt actually is, because UniProt consists of a couple of different databases. So there's this really great guide on UniProt. Typically, when you're dealing with UniProt, you're going to be dealing with UniProt KB, and that's the UniProt knowledge base. There's also UniParc and UniRef. UniParc is the sequence archive, and it has like all sorts of sequences, including both obsolete sequences and current sequences. UniRef is really interesting, so it's great if you want to do a blast to find sequences that are similar, but you want to weed out sequences that are identical. And so UniRef 50, it's going to be sequences that are 50% identical. The 90 and 100, what it does is it takes those sequences that are really similar and it puts them into clusters. So then you're searching against the clusters instead of searching against the individual sequences, and you can kind of find more distant relationships. And so not going to go into UniRef. Let's focus on UniProt KB. That's what you're typically going to be using. When you're dealing with UniProt KB, there's going to be two different databases within it. And these are going to be the SwissProt and Trumbull. Now, the SwissProt is going to be the sequences that are manually curated. So they're going to have a lot of really nice annotations. So you'll see information about various processing, variants, mutagenesis sites, cellular location, all this great stuff. And you'll see there's going to be a lot of these citations where like people actually found these in publications. So you can see that some of these entries are going to have a really high annotation score, indicating that there's a lot of manual curation that goes into these, people actually going through and adding all this information. You can see that this is in the review, the SwissProt, and it has this gold symbol. The other database is Trumbull. And so Tremble is sequences that haven't been manually curated. They've more been computationally assigned. So they say, okay, well, this protein, we haven't actually experimented with it, but the sequence of it is really, really similar to the sequence of malodehydrogenase in these other bacterial species. So much so that we can kind of predict even where binding sites will be. But all of that information is just going to be predicted, and not that it's not going to be actually, we don't have the physical evidence for it. And so that's not going to, and it's not manually curated. So people haven't actually gone through this entry and added all this experimental data and stuff. So it's going to be in this Tremble. This is unreviewed. This is one of these unreviewed entries. And so typically you want to use those with a little more caution. Each protein in both Tremble and SwissProt will have its own entry page. It'll have its own identifier, unique identifier, and unique name. And these are all going to show up in the FASTA code format for this in the header bar. To actually download that FASTA format, what you can do is when you're on an entry, if you go to sequence, so you can click on it from here, or you can scroll down, you'll see the sequence here. If you click download, it's a little confusing because it takes you to a new window and you're like, I thought that we would download something, but you just need to right click and save as, and you can save it as a FASTA format, just like a text file. It'll save it with the .fasta ending. So that's one way you could get it. And I could do that for any entries that I want. And so I could go ahead and I could do that here too and get the one for Bacillus subtilis malodehydrogenase. Another way that I could get this is if I go to my cart. So I have these added to my cart. I could select the sequences that I want. I could go to download, and it would allow me to download the FASTA format. I can, if I have a lot of these, I can download as many as I want. I don't know, actually, it might have a limit, but I can download these and I can do it compressed or not compressed. You could also have it show you the preview so you can see what it'll look like, and then you could go ahead and download it. So if I went ahead and downloaded it, what I would find was a sequence like this that I could open with a plain text editor. So let's take a look at this header bar in a little more detail. On top of here, I have the malodehydrogenase sequence from Bacillus offensis, and then below from Bacillus subtilis. I can tell where one entry starts based on where the carat is. So this vertical pipe, this is going to be, remember, telling us about sort of database information. And so the first two letters that we have when we have a UniProt sequence, this is going to be the database. So with UniProt KB, remember, we have those two databases. We have SwissProt and we have Tremble. SwissProt has the manually curated information, and then Tremble has the more auto-curated information. So SP, this is telling us this is from the SwissProt, and TR, that's telling us this is from the Tremble. Both of these will be followed by the vertical pipe, which separates it from the unique identifier. So we see those unique identifiers here. And if we were to go and we were to actually look at the entry, this corresponds to that number that we saw up here. So this AOAO thingamabob, that's what we're seeing right here. And then this part that's after the second pipe, that's going to be the second. And so this was our unique identifier, and this was going to be our entry name. And so those are going to be different for each of the things that you have. Now we're going to have the gene name, though, and you can see that this is the same for both of these because it's the same gene, but it's in different species. What species, you might ask? Well, that's where we see the OS. So the OS is going to tell us the organism name. So here it's Bacillus offensis, and here it's Bacillus subtilis. It's a little more specific here and telling us that it's Bacillus subtilis from strain 168. So with bacteria, you'll sometimes see things like a strain. Now OX, this is the organism identifier, and so it's a number code that stands for the organism. Then you have GN, that's going to be the gene name. That's going to be optional, so you won't always see this. Sometimes you won't even know what the gene does. You'll just have this sort of sequence, and so then you might not have that GN. PE, protein existence. This one's really important to know about. So protein existence, sometimes we have the sequences for proteins, or we compute the sequences for a protein. We say we have this, we did the sequencing information of the genetic sequences. And we see that there's this place, and it looks like it has a gene that should make something that resembles malate dehydrogenase. We don't know if it actually makes malate dehydrogenase, but it seems like it should make malate dehydrogenase. And so we're going to predict that it makes malate dehydrogenase, and we're just going to assign it to malate dehydrogenase. But we don't know that it's actually malate dehydrogenase, and we don't know that it's actually made. So that sort of information can go into these databases, but then they won't have very high protein evidence. Sometimes you also see things like if alternate isoforms where they start at different start sites and then they might have these really short peptides and you don't know if they're actually made. So those would have things like protein uncertain. So we give these proteins a protein existence score from 1 to 5, 1 being the best and 5 being the worst. And best and worst, that just means the most evidence versus the least evidence that this protein actually exists. So if you have a PE score of 1, that's saying we have experimental evidence at the protein level. So maybe someone's done a Western blot or something, and they actually have been able to detect this protein is actually being made in cells. If you have a 2, you have experimental evidence at the transcript level. So we saw the mRNA, but we didn't see the actual protein. And then 3, we say, okay, so we have a 3 here, and that's saying that we have protein inferred from homology. So this protein is really, really, really similar to this protein, and so we're just going to go ahead and say that this is probably this protein, but we haven't actually seen this protein. We haven't seen the mRNA for this protein, but it's so, so, so similar to this other one that we're pretty confident that it exists. And then 4 and 5, these are going to be a bit less certain, with 5 being the most uncertain that it actually exists. Now, sometimes there's just a lack of evidence because these are sequences that people have not looked into very much. And so often these like sequences in Trumbull, these are going to be organisms that people haven't researched as much. And so there's not as much manual curation because people just haven't done the experiments to work with them. And so you're not going to have the evidence of the protein because no one's actually worked with these. It doesn't mean that the protein doesn't exist. So if you have a lower protein evidence score, it doesn't necessarily mean that the protein doesn't exist. I'd be more suspicious if you had a lower protein evidence score on a species like a human, where people have done lots and lots of research, and that still doesn't mean it doesn't exist, but it's more likely that if people have done a lot of searching but they haven't found it, it might not be there, compared to if it really seems like it should be there, but we haven't looked there, then that would be more understandable why you have a lower protein evidence score. So those are just some kind of things to keep in mind. Let's take a look at comparing some sequences. And so I have a lot of sequences in my cart of malate dehydrogenase from different species. And what I did was I went ahead and I did that download, and I downloaded all of these. And so you can see if I do a preview, I'll see something like this. But what I did was I actually downloaded the file and opened it in my text editor. And so what I see is something like this. So remember that you're still, for all of these, the first 2 is going to be that database it came from, so Trumbull or SwissProt. And so you can see that I have a sort of variation of these. Some of these are from the SwissProt and some of these are from Trumbull. The ones from the more common species, E. coli, subtilis, humans, are going to be from the SwissProt. And then some of these from like Safensis, from these other strains of bacteria that aren't that well studied, you'll see that these are coming from the Trumbull. You see here that I have multiple sequences from Bacillus safensis. So here I have malate dehydrogenase. I have the lactate dehydrogenase. I have a couple lactate dehydrogenases. All these are going to have the same OS, the same organism name, and the same OX, so the organism identifier, so that number, because they're the same species. But they'll have different gene names, and they'll also have potentially different protein evidence, as well as different sequence versions. These are all kind of have these protein evidence of 3, saying that they are going to have protein inferred from homology because they're really similar to proteins, say, from subtilis, which actually is in that SwissProt, but we don't have the physical evidence from Safensis. If they go to a different species, here you'll see that they have different organism names. They'll have different organism identifiers. They might have the same gene name, though. And so that's kind of how you evaluate these codes up here. That's just going to have information. The information for sequences that are isoforms are going to be slightly different. I'll point you to the UniProt, which has a really great guide on this with more information. The basic difference when you're dealing with isoforms is that you're not going to have the protein existence information because that relies in part on cDNA copies, which you can't really tell apart for the isoforms, what protein's being made from the cDNA and stuff. And cDNA, that's just the DNA version of the mRNA. So basically transcript information, you can't really tell apart some of the isoforms, so they don't try to say whether or not there's strong existence for a specific isoform. But what it will do is it'll give you the isoform sequence. So it'll be slightly different from the canonical sequence, the main sequence. And in the accession number, you'll see that it has a dash after its name. So for example, if I went to this malic dehydrogenase human, which is where these are coming from, if I go to sequence and isoform, it'll identify one as the canonical sequence, so the main sequence. And if you were to just download the FASTA format, it would give you the format for this sequence of this canonical format. But there's also these alternative isoforms. And so if I were to have the alternative isoforms, I could download these individually, or I could add them both to my cart. And they're already in my cart, but I would add them to my cart with the little cart symbol, and then I could go ahead and download them, which is what I did before, which got me to this thing like this. Finally, let's talk a little bit about how we can change these to make things easier to work with. So those headers are really great if you want all that information, but sometimes they can get in the way. An example of this is when you're trying to do an alignment. So say I want to align the Bacillus safensis and Bacillus subtilis malic dehydrogenase. You can see that they kind of have these names that are not very informative about what they actually are. But I'm going to go ahead and select these. I'm going to go to Tools. I'm going to align them. What you'll see now is I'll get a window like this. To start, I'm just going to have it run default, but you can see that it entered the FASTA format of these files. So I have the caretted bars with the headers for these different things, and then they're each separated by a caret. And the first line, the header line, has no spaces, but then the sequences are going to be broken up into these 60 to 80 character lines. I'm just going to go ahead and run the align as is. It's completed. I'm going to go ahead and click on this. And now I'll see the alignment, and it'll look something like this. Now I can kind of tell that this one's subtilis malic dehydrogenase, but if I hadn't worked with this so much, I would not know that that's Bacillus safensis malic dehydrogenase. Definitely not very clear to see what's what. And especially if that bar were to be smaller, such as when you put it in some other software, this will actually look even smaller. It'll cut it off even more, and it's really hard to tell what's what. So what I'm going to do is I'm actually going to change that header line. Rather than calling this whole long thing, what I really want to know is this is the malic dehydrogenase from Bacillus safensis. So I'm just going to call this bsaf underscore MDH. And I'm going to change this one to just be bsub MDH. Now if I can go ahead and run the align, completed. So I'm going to go ahead and click on that. Much easier to tell what's what. Because FASTA formats are just plain text files, I can also just edit it in the text file itself and then upload the text file to whatever tool I wanted, such as if I wanted to do an alignment, I could load from a text file here, or I could just copy and paste, but you'll see that it wants things in FASTA format, and now you know how to make a thing into FASTA format. All you gotta do is take the sequence of a protein or nucleic acid, ensure that it is in that proper one-letter IUPAC format where the amino acids have their conventional one-letter abbreviations. Yes, it's really important you learn those abbreviations, and this is one of the reasons why. And so then you also have the wildcard, so you'll have an N for any nucleotide, you'll have an X for any amino acid, an asterisk is going to be a stop codon in a translated amino acid, a dash is going to be a gap of indeterminate length in either a protein or a nucleic acid. There's some other wildcards in nucleic acids, and so I'll point you to a guide. Split those sequence up into the 60 to 80 characters per line, and separate your different sequences with each of them has their own caretted header line. When you download these sequences from a database, the various places you'll find how to download the sequence. And so in UniProt, you can go to sequence, download it directly from there, or download it from your cart. You can download multiple from your cart. You can go ahead and then look at the header lines to get more information about the species, about the organism. This is really great information if you're doing some computational work. This information is really helpful for computers that are trying to, say, sort things by their species or by their gene name. But if you don't want that information, just change the header line, make it something that is more useful for you. So, hope that helped you understand the FASTA format. Really not that bad and really helpful for a lot of different things.